Hello there. My name is Connor Kennedy. Today is October 19th, 2020, and I will be presenting to you the determination of gender, genes versus hormones. Alright, so let's jump into it. Sex determination. Sex organs consist of three structures: the gonads, which produce gametes from eggs and sperm to form individuals. The testes produce sperm, and then we see the ovaries produce eggs. For the internal genitalia, we see accessory glands and ducts, which these connect gonads to the outside environment. And external genitalia, this is a group of all external reproductive structures. And with sexual development programmed in the human genome, we see nucleated cells contain 46 chromosomes, but the egg and sperm contain 23 chromosomes. There are two sex chromosomes, X and Y. These contain genes to direct individuals to produce internal and external sex organs. X is larger than Y and contains many genes that Y does not have. The egg and sperm are haploid, but when they come together, they form a zygote. This creates a unique set of chromosomes from the father and the mother. And next, we talk about genetic aspects of sex determination. The sex chromosomes inherited determine the genetic sex. We see that two X chromosomes give us a female, and an X and a Y chromosome gives us a male. The X chromosome comes from the mother, and we see the Y chromosome come from the father. If an abnormal distribution of sex chromosomes happens at fertilization, then the absence or presence of Y will determine gender sex. For a female fetus, when ovaries develop, there will be an inactivation of one of the X chromosomes, and either the maternal or the paternal X chromosome will become the active X chromosome. For sexual differentiation in early development. Not until 7th week it becomes more possible to determine sex morphologically in embryonic development. Before that point, this is called bipotential embryonic development. With the bipotential, we see that the gonad has an outer cortex and an inner medulla. With the appropriate signal, the medulla will develop into testes, and without this signal, the cortex will become ovarian tissue. The bipotential internal genitalia has two accessory ducts, the Wolferian ducts and the Mullerian ducts. As we see development proceed, one of the ducts will degenerate and the other will develop depending on male or female development. Also, bipotential external genitalia consists of genital tubercle, urethral folds, urethral groove, and lagosacrotal swellings. There is the SRY gene. This is the sex-determining region of the Y chromosome. The presence of this gene, we see the gonads develop into testes, and without this gene, we see the gonads develop into ovaries, along with help from female-specific genes as well. And to be more specific, we talk about the male embryonic development next. Here we start to see hormones take play. The SRY gene produces a protein called the testes determining factor, which binds to DNA activating other genes to help direct development of gonadal testes. Once testes are recognizable, they begin to secrete specific hormones. The testicular Sertoli cells, they secrete glycoprotein anti-Mullerian hormone. The testicular interstitial Leydig cells secrete androgens. This includes testosterone and dihydrotestosterone. The anti-Mullerian hormone causes the embryonic Mullerian ducts to regress, and the testosterone turns Wolferian ducts into male structures, which include the epididymis, vas deferens, and a seminal vesicle. The testosterone controls migration of testes from the abdomen to the scrotum, and the dihydrotestosterone controls the characteristics of the remaining external genitalia. And if we focus more in the picture on the right, we're looking more on the left side of that picture. Now for female embryonic development. And we see the same picture here in this slide as well, but now we're focusing more on the right side in the pink area. So the female embryonic development is almost the opposite as the male development, except it is seen to be more extremely complex compared to the male. We do not see the SRY gene present in female development. Female development is extremely complex because it involves multiple genes for development, and it needs functional ovaries. When the anti-Mullerian hormone is absent, we will see portions of the upper vagina, the uterus, and the fallopian tube start to develop. And same with testosterone and dihydrotestosterone. When these two are absent, we will see the Wolferian ducts degenerate, and this will bring on female characteristics of the external genitalia. And this is my sources, and I want to thank you for watching my presentation on the determination of gender, genes versus hormones.